Hello and welcome to BioBiz Buzz. I'm your host, Mike Ward, and I'm the Global Head of Life Sciences and Healthcare Thought Leadership at Clarivate, which is a multinational provider of data, insights, and intelligence that helps healthcare companies accelerate their innovation. In this edition of BioBiz Buzz, we're going to envision a future where cardiovascular diseases are addressed not merely by managing symptoms, but actually by reprogramming the fundamental mechanisms that cause them. We're going to explore the groundbreaking research work of the Swedish biotech company Cereno Scientific, which is a pioneer in harnessing epigenetic modulation. It's a technology that has the potential to transform drug development in the pharmaceutical industry. At its core, epigenetic modulation adjusts gene expression without actually changing the DNA itself, so providing a reversible and precise method to actually combat disease. While this approach has already sort of been transforming oncology, Cereno is actually leading the way in applying it to cardiovascular diseases, which is a field that is exactly ripe for innovation. The company's primary candidates, CS1 and CS014, are histone deacetylase HDACi inhibitors, which are designed to reverse pathological remodeling, reduce inflammation, and prevent thrombosis, all while actually avoiding the traditional side effects, such as bleeding risks. So stay tuned as we explore how scientific advances are reshaping drug discovery one gene switch at a time. And to do this, I'm delighted to be joined by Sten Sörensen, who is the CEO of Cereno Scientific, the clinical biotech company that is making significant strides in rare disease therapeutics. Thanks for joining me, Sten.
Thank you, Mike. It's a pleasure to be here with you.
So Cereno recently pivoted to focus exclusively on rare diseases. So what actually drove this strategic shift?
Well, it's really the unique advantages of the rare disease space, having shorter timelines, less capital-intensive development, and in many times large unserved need in the patient categories that are affected by rare diseases. But fundamentally, the reason we moved to rare diseases is that we saw a very good fit, like a hand-in-a-glove fit for our lead project, and actually our two programs with the epigenetic modulators, HDACi inhibitors CS1 and CS014. So it's a combination of our technology and unique mode of action and programs and the space of rare disease for development.
Yeah, so actually, I already mentioned in my intro about epigenetic modulation. I think it would be very helpful if you could sort of, explain the underlying science of Cereno Scientific's lead programs.
Yeah, sure. So the cardiovascular space has been, in a way, neglected for a couple of decades, and a lot of focus have been on oncology, despite the very fact that most humans die of cardiovascular diseases. The very most of thrombosis, actually, or such as stroke, myocardial infarction, and other implications of stroke. And the company was formed on the basis of the vision to develop a drug that could prevent thrombosis without the serious risk of the current drugs of bleeding, which are fatal in about 3 to 4% of the patients and affect 25% of the patients in clinical programs. So the very high need of preventing thrombosis with needs for more efficacy but less side effects of this bleeding risk propelled the Professor Sörensen to develop the company and the idea of epigenetic modulation. And the company was in search for a mode of action that could deliver epigenetic modulation, increase actually the synthesis of a protein that's essential for preventing and lowering the risk of thrombosis that we have as a defense mechanism in our body. That protein is called TPA. And the company found VPA, valproic acid, to be a prominent HDACi inhibitor. And with this HDACi inhibition, you can actually increase synthesis, storage, and release capacity of TPA. So that's how the journey for Cereno started. When I joined and reconnected with our current scientific advisor board and this chairman, Dr. Bertram Pitt, Professor Emeritus from the University of Michigan, we discussed and agreed to broaden the investigation and evaluation of the HDAC inhibitors with VPA. And we should know here that VPA has been in man for neurological diseases and disorders for over 60 years. So the fact that it's an HDAC inhibitor, an epigenetic modulator, was discovered maybe 20 years ago. And a lot of research has been done with various research groups around the world and published that has showed that this epigenetic mode of action with VPA and HDAC inhibition has documented the ability to lower systemic and pulmonary arterial pressure, the ability to be antifibrotic, anti-inflammatory, and also to prevent vascular and heart remodeling, and in addition to reverse it. And the last characteristic is actually then to be able to prevent thrombosis without increasing bleeding risk. This is all published information. We found up to almost 600 articles on HDAC inhibition, most of them VPA. So that prompted our pivot to search for a disease that would be our lead indication, moving away from thrombosis as the more general approach. And we defined pulmonary arterial hypertension as a disease that is severely underserved of the drugs available today, and where these characteristics fit very well with the pathological drivers of the disease, of the very progressive vascular remodeling, leading to right heart failure and death within 7 years, mostly affecting females.
So with CS1, clearly there are already existing therapies. So how do you differentiate what you're developing compared with what's already in place?
Yes, so a very good question. So the selection of PAH, pulmonary arterial hypertension, as the lead indication for this unique mode of action, and with these characteristics already documented as proof of concept in various animal models, including pulmonary arterial hypertension, had shown in studies on rat models in PAH the ability to both prevent vascular and cardiac remodeling and also reverse it, in addition to lowering the pulmonary pressure and in addition to reduce the fibrotic content of the vascular and the heart. This is exactly what happens in these patients. However, as with left heart failure, classical heart failure, if you will, originally these diseases, heart failure has been treated as a pump and plumbing effort. So originally inotropic agents and diuretic agents were used in left heart failure. I have myself been involved in paradigm shifting that into using more hormonal control drugs, such as the MR antagonists with the RALS trial that was initiated together with Dr. Bertram Pitt, who was principal investigator at that time, published in New England Journal of Medicine in 1999. And going back to AstraZeneca in the mid-90s, I, together with Gunnar Olsson, who's also on our board, initiated the MERIT-HF trial with a beta blocker, who, by the way, was contraindicated at the time for heart failure. But both of these trials improved survival and quality of life, survival with more than 30%, published in New England Journal of Medicine. So that started in the early 90s, this movement with more agents for left heart failure. Now there are 6 to 7 agents. However, in pulmonary arterial hypertension, which is a progressive narrowing of the muscle in the pulmonary artery, leading to higher pressure and more workload on the right heart, eventually leading to it failing, so right heart failure. The development of drugs, more than pump and plumbing, is 30 years behind left heart failure, in our opinion. And so we are moving here into the right heart failures field with a unique mode of action addressing the underlying progressive vascular remodeling. The current agents are vasodilators, so they're still there, dilating the vessel, reducing the pressure, helping the patients. And we shouldn't neglect the fact that the patient has a life expectancy have increased from around 2.5 to 7 years mean with these agents. So they are very helpful initially in the disease. However, they do not address the root cause of the disease and the pathological progression of vascular and heart remodeling. That's where we come in, and that's where the research field now is focusing. So we have seen in our preclinical work, as I mentioned, the ability to do so. So prevent and reverse pulmonary, arterial, and cardiac remodeling, and also lowering the pressure. And in addition, lowering the fibrotic content of the vessels. Now, we have then moved our HDAC inhibitor, repurposed VPA from neurological disorders into the cardiovascular space with a new formulation, orphan drug designation, and a patent portfolio of 3 patent families. So we've moved that in and just completed a phase 2A in the pulmonary arterial patient population. And we saw some really interesting and positive data on disease modification as we deemed the results show. And I can relate to you that we have seen in our study, which was in a fairly small group of patients, 25 patients, but we saw from baseline an improvement of the risk of mortality over this 3 months period where we added our drug to standard of care. So we saw a reduction of prognosis, risk of mortality. We saw an improvement of the New York Heart Functional Class population to lighter functional class in 1/3 of the patients. And we saw also improvement of quality of life. And maybe most interestingly, we saw an improvement of the right heart to pump better, measured as an improvement of the global longitudinal strain. So we have seen here, adding our new mode of action to vasodilator standard of care, that we can move functional class, we can reduce prognosis, we see patients feeling better, and their heart is pumping better.
Right. So you mentioned that you've done the phase 2A. So, what are the prospects for phase 2B, phase 3 with CS1?
Well, yeah, so phase 2A, of course, is the primary endpoint for safety and tolerance. And we were quite confident that our drug would not show any serious side effects and not more than what has been seen with non-serious side effects using it in neurological disorders. We are actually using this drug in much lower dosages than has been used previously for other indications. So what we really were after in our phase 2A trial was signals of disease-modifying capacity and signals of helping the patients. And so we just completed our Type C meeting with the FDA this Monday, and we are now aligned with the FDA in our ambitions to move forward and study our drug on top of standard of care in a wider audience and a placebo-controlled trial. So there we aim to confirm our efficacy for these patients on top of standard of care.
And the other sort of like lead program that you have, CS014, I understand that that's sort of been focused on IPF. Why are you moving into that space?
Well, the reason is really that if you have a mode of action that has documented the characteristics that I mentioned up front here in our talk, so the ability to lower systemic pressure, the ability to act as an anti-inflammatory agent, the ability to prevent fibrotic development, the ability to reverse muscle, vascular remodeling, as well as cardiac remodeling, and the ability to prevent thrombosis without increasing bleeds. If you have those characteristics documented in preclinical work, you would love to explore this for patients that are suffering from cardiovascular and pulmonary diseases in various indications. So we at Cereno and our collaborators view our HDAC inhibitor vision as a platform to be utilized for various indications. We, as a smaller biotech, are benefiting of focusing on rare diseases, as I mentioned up front, because of the less capital intense and the high unmet need. But we believe that this platform can be explored in a wider range of indications. And that's also why we, 5 years ago, defined the vision to develop a new chemical entity with the similar characteristics as the original drug CS1, but with a better safety profile and a longer protection for its new chemical entity. And that's CS014. It's actually a deuterized version of EPA. So the choice of moving into defining IPF as our initial target indication for CS014 was, in a way, obvious for us because we have seen there are documentation of VPA in IPF models, so proof of concept models that in preclinical, and we have also documented dose-dependent impact on fibrosis and vascularization in pH models with CS014. So we believe that drug can be very useful in IPF and other lung indications.
To develop these candidates, it's not an inexpensive process. So do you have the financial firepower to do this yourself, or are you looking for pharmaceutical partners to assist you?
Yeah, it's a good question. You know, I've been working with Cereno since it was an idea, more or less. We had no patents, but we had a vision together, and we saw the space, and we have capitalized the company. I've been with the company for 10 years, and we capitalized it on the smaller Swedish market. And at the time that we started, there wasn't really venture capital available for ideas, more for companies that already had patents or some proof of concept somewhere. So we went to the open stock market in Sweden for smaller biotech ventures and raised our first capital. And we have continued to work with the stock market here, and we uplifted to NASDAQ First North last year. And we have 10,000 shareholders, retail in Sweden. And of course, when you move towards phase 2B or later clinical stage development, the stakes gets higher, you need more capital. So we are looking to eventually finance with the higher level venture capital or funds as one avenue of financing our later stage programs. Another avenue is, of course, partnering. So we are in talks with various pharmaceutical companies internationally for potential collaboration. But we are quite opportunistic when it comes to the current stage that we are in. The retail market has been quite tough for biotechs since post-COVID, since 21. I think you are well aware. So to do share issues on the market has been afflicted with quite significant rebates. So we decided to, in 2 instances, actually do a deal with Fenja Capital first, and then with Fenja Capital and their partner Arena, the US partner, to move the company past significant milestones, which we believed would be a good model for our company in relation to benefit the shareholders best, I would say, and the company. So avoid significant dilution whilst moving past significant milestones. And I believe we are on our way to do so. We are in the middle. And I think also we have seen lately that the market is responding to us reaching these milestones.
So, what specific milestones should we be watching out for from Cereno in 2025?
Well, I think when we were in 24, we said we would conclude the phase 2A trial and communicate our findings. And we did that in the beginning of February. So that part we completed already this year. And we saw these very nice signals of efficacy of disease modification and helping the patients in PH. And then our key milestones for the CS1 program this year is to align with the FDA and get an agreement and eventually an acceptance of an IND to move forward and initiate that trial. So a phase 2B trial with CS1 in PH. And we just, as I mentioned, had the Type C meeting where we are in alignment with our thinking, with what the FDA views on our thinking of such a protocol and ambition. So we aim to get that IND accepted this year and initiate the trial next year. But we have other things going on with CS1. And actually, the investigators in our trial requested us to apply for compassionate use or extended access program to the FDA because they didn't want to let go of our drug based on their experience with adding it to standard of care in the 3-month trial. So we did so the day before Christmas Eve. We applied to FDA and got it approved by the end of January last year. And we have as many as 50% of our patients now on long-term clinical use of CS1 and PH on top of standard of care. So we have now upcoming evaluation at 3-4 months readouts from the patient group. So that will be very interesting to see, the long-term use here, 4, 8, and 12 months on. And those patients went in on therapy in Q3 and Q4 last year. So that's one important readout. Another one is that we, based on what we have seen in preclinical and our experience of the trial, we signed a collaboration agreement with Fluidda, who has a very interesting technology, a CT scan technology, where you can CT scan the patient and look for impact clinically on disease modification on the small pulmonary arteries in these patients. So we are studying with Fluidda technology, and we'll also expect to get a readout of that. That readout will come after the 12 months. So IND acceptance, readout of expanded access, and then Fluidda technology going on. With the second program, CS014, the new chemical entity, we actually a week ago completed the last patient last visit. So that study, phase 1, is going to deliver top line in June from that study. So then we have 2 programs actually moving into phase 2B. And CS014 then is defined to move to IPF. So we expect an IND acceptance also this year for CS014.
So it sounds like you've got a very busy year.
Yeah.
So, well, Sten, thanks very much for sharing Cereno's visionary approach. What you're doing clearly exemplifies the power of targeted innovation. So I'll be following your progress very, very closely. So I'm curious how you, the audience, felt about this conversation. You can send your responses to biobizbuzz.com. And if you want to stay tuned for more editions of BioBiz Buzz, follow our LinkedIn page where we'll be posting alerts for future episode releases. So in closing, I'd like to thank Sten again for joining me. I'm Mike Ward, and I hope you join me for the next episode of BioBiz Buzz. Goodbye.
Thanks, Mike. Thanks for having me.